It was September 3rd, 1928. A Scottish scientist sat down at his laboratory bench at St. Mary's Hospital in London, exhausted and emotionally drained after returning from a 2-week holiday with his family in Suffolk. He had spent years conducting meticulous research on Staphylococcus bacteria, convinced that somewhere in the microbial world existed something powerful enough to stop bacterial infections. His name was Alexander Fleming, and on this particular morning, tired and ready to give up on his years of failed experiments, he had absolutely no idea that in the next 15 minutes, he would stumble onto a discovery that would save more lives than any invention in human history. But here's the thing that makes this story truly remarkable and deeply unsettling. He almost threw it away. He almost discarded the most important petri dish of his entire life. By the time Alexander Fleming discovered what was actually growing inside that contaminated plate, millions of soldiers were already dead from infections that would later be completely preventable. Thousands of mothers had died during childbirth from blood poisoning. Children had been buried because a simple cut had turned into a fatal infection. But nobody knew yet, absolutely nobody could have possibly known, that salvation was inches away from being lost forever. That a moment of curiosity, or perhaps a moment of carelessness and waste, would determine the fate of millions of human beings. Before we dive deeper, let us know in the comments where you're watching from. We'd love to hear from you, and don't forget to hit that subscribe button so you never miss any of our upcoming videos. Before we go any further into this astonishing story, I need you to understand 3 critical questions that shaped the next decade of Alexander Fleming's life, and ultimately the course of modern medicine itself. First, what exactly was that strange, unidentifiable mold that had contaminated his Staphylococcus culture while he was away on holiday? Second, why did this mysterious fungus possess the almost supernatural ability to kill every single bacterium surrounding it, as if it were an invisible assassin working methodically through the petri dish? And the third question, the one that matters most to the millions of people walking around alive today because of what happened next, could this mold actually be harnessed to save the world, or would political obstacles, wartime destruction, chemical instability, and human misunderstanding bury this discovery so deeply that it would be lost to history forever? These 3 questions consumed Fleming's thoughts. These questions drove him forward when conventional science told him he was chasing ghosts. And these questions would eventually pull 3 other brilliant scientists into the most important scientific collaboration of the 20th century. Because Fleming did not work alone. He was merely the first to see. The others would dedicate themselves to believing. Alexander Fleming was not born a genius destined to revolutionize medicine. He was born into struggle, born into a family fighting against poverty and obscurity. His father was a Scottish hill farmer, a man who worked the land in Darvel, Ayrshire, a remote and harsh part of Scotland. Fleming entered the world on August 6, 1881, the 7th of 8 children in his family. His father was not a gentle man, and he died when young Alexander was only 7 years old, leaving his mother to raise 7 children alone in a landscape that offered little mercy to the weak or unprepared. But Fleming's mother was formidable. She refused to allow her children to accept the limitations of farm life. She pushed them relentlessly toward education, toward opportunity, toward something better than what their circumstances suggested was possible. Fleming's older siblings became doctors and teachers. Fleming himself became determined to escape the farms and fields forever. He studied sciences with the intensity of someone who understood that education was his only weapon against poverty. By the time he was old enough to attend university, he was already different from other students. He had witnessed the desperation of poverty. He had seen what sickness could do to families with no resources to fight back. He had developed, without even realizing it, an almost obsessive need to find solutions to medical problems that devastated ordinary people. In the 1920s, Alexander Fleming had become a respected bacteriologist at St. Mary's Hospital in London. He was no longer that struggling farm boy fighting against poverty. He had built a reputation as a meticulous researcher, a man who paid extraordinary attention to detail, and most importantly, a man who asked questions that other scientists considered too radical or unconventional. But by 1928, Fleming was facing a personal crisis that no amount of education or scientific achievement could completely resolve. He was frustrated. He was obsessed with a problem that seemed completely unsolvable. Every day at St. Mary's, he witnessed the devastating reality of bacterial infections. He saw patients arrive at the hospital with what seemed like simple wounds, cuts, scratches, minor injuries that should have been trivial. He watched those same patients deteriorate over days and weeks as their bodies were consumed from the inside by bacterial infections that no medicine could stop. He watched people die from gonorrhea. He watched mothers die from childbirth fever. He watched soldiers arrive from World War I with infected wounds that resulted in amputation or death. In 1922, Fleming had actually discovered something called lysozyme, an enzyme found in tears and saliva that possessed some antibacterial properties. That discovery had proven he was capable of breakthrough work. But Fleming knew, with the kind of absolute certainty that comes from understanding the stakes involved, that lysozyme was not powerful enough to save lives on a meaningful scale. There had to be something stronger. There had to be something capable of killing the bacteria that destroyed human bodies without simultaneously destroying the human tissue itself. That was the obsession that consumed Fleming every single day in 1928. That was the question keeping him awake at night. That was the reason he inoculated those particular Staphylococcus culture plates before he left for his holiday. He was testing variations in bacterial growth, searching for patterns, searching for anything that might lead to a breakthrough. He had no idea what he was about to discover. Fleming returned to his laboratory on September 3, 1928, not expecting anything unusual. He had been away for 2 weeks. His research scholar, Stuart Craddock, had continued working in his absence, but Fleming had given instructions that the laboratory should be cleaned and prepared for his return. He walked into his laboratory like he had walked into it thousands of times before. He sorted through his petri dishes methodically, checking on cultures he had left behind, making notes about which ones had developed properly, and which ones had been lost to contamination or failed to grow as expected. Most of the plates looked normal. Most of them represented either successful experiments or expected failures. But then Fleming picked up one particular plate, and something about it stopped him cold. The plate contained a culture of Staphylococcus bacteria, and it had been contaminated with a mold. That alone would normally be a disaster. Contamination meant the experiment was ruined. It meant throwing the plate away and starting over. It meant frustration and wasted time. Most scientists in Fleming's position would have immediately discarded the plate without a second thought. But Fleming looked closer. He examined the plate more carefully, and what he saw made him stop everything he was doing. Around the mold, there was a clear zone. All around the fungal growth, the Staphylococcus bacteria had been completely dissolved. The bacteria immediately adjacent to the mold had been completely destroyed. The bacteria further away were still present and normal, but directly around the mold, there was nothing but empty space where living bacteria should have been. The colonies had become transparent. They were obviously undergoing lysis, the process by which bacterial cell walls rupture and the cell dies. Fleming reportedly looked at the plate and said something so simple, so understated, that it has become legendary in the history of science. He said, "That's funny." But in that moment, Fleming's entire perspective shifted. This was not contamination. This was a revelation. This was nature showing him something that decades of deliberate experimentation had failed to produce. He showed the contaminated plate to his colleague, Merlin Price, who had actually worked with Fleming on his earlier lysozyme research. Price took one look at the plate and said something that was meant to be encouraging but must have struck Fleming as almost comically inadequate in the face of what he was seeing. Price said, "That's how you discovered lysozyme." But Fleming knew instinctively that this was different. This was more powerful. This was more significant. This was something unprecedented. Fleming decided immediately that he would investigate this mold. He identified it, working with his colleague Charles Latouche, determining that it belonged to the genus Penicillium. Initially, Latouche believed it was Penicillium rubrum, but eventually it would be corrected and properly identified as Penicillium notatum, a rare variant that would later become known as Penicillium chrysogenum. But at that moment, Fleming did not care about the precise classification. He cared about what the mold was doing to the bacteria. For the next 3 days and nights, Fleming existed in a state of almost feverish focus. He barely slept. He barely left the laboratory. He performed experiment after experiment, testing the properties of this mold, measuring the zone of bacterial destruction, attempting to understand the mechanism by which the mold was killing the bacteria. Every single test he conducted produced the same extraordinary result. The mold was secreting something, some chemical compound that was lethal to Staphylococcus and many other gram-positive bacteria. But this lethal substance was not toxic to the mold itself, which continued to grow and flourish. This was not a situation where one organism was simply overwhelming another in a battle for resources. This was one organism deliberately attacking another while remaining completely unaffected by its own weapon. Fleming grew the mold in pure culture. He prepared culture broth, containing only the mold and no bacteria. He found that the broth possessed remarkable antibacterial properties. He could filter out the mold, and the remaining liquid still killed bacteria. This meant that the mold was producing a discrete chemical substance, a substance that could be separated from the mold itself. He called this substance penicillin. He explained his choice of name by saying simply that he wanted to avoid the constant, cumbersome repetition of saying mold juice, which is what he and his colleagues had initially called the substance. Penicillin was shorter. It was more scientific. It was more elegant. And Fleming was convinced that he had stumbled onto something absolutely revolutionary. Fleming published his findings in the British Journal of Experimental Pathology in June of 1929. He sent out his paper to the scientific community. He presented his findings to colleagues. He demonstrated the penicillin's extraordinary antibacterial properties to anyone willing to listen. And then something happened that would define the next decade of Fleming's life and the lives of millions of people around the world. Nothing. Absolutely nothing happened. The scientific community responded to Fleming's discovery with indifference. Some scientists showed mild interest. A few requested samples of the penicillin mold, but the dominant response was skepticism or, worse, simple disregard. Physicians did not believe that penicillin could be developed into a practical medicine. Pharmaceutical companies saw no profit in attempting to purify and mass-produce a substance from a mold. The chemical industry was not interested in investing resources into something so unpredictable and unstable. Fleming found that penicillin was extraordinarily difficult to extract from the mold broth in any usable form. The substance was chemically unstable. It degraded rapidly if exposed to heat. It broke down if the pH of the solution became too acidic or too alkaline. It seemed almost deliberately resistant to purification. Fleming worked with research scholars Stuart Craddock and Frederick Ridley in his laboratory, attempting to develop methods for isolating penicillin in concentrated form. Their progress was painfully slow. They could produce only tiny quantities of a crude penicillin solution. There was no way, using 1920s and 1930s chemistry, to create enough penicillin for any meaningful medical application. By 1931, Fleming had essentially abandoned his penicillin research. He had other projects demanding his attention. He had duties as a bacteriologist at the hospital. He had teaching responsibilities. The penicillin discovery was relegated to his files, filed away as an interesting scientific curiosity that had no practical application. And there it remained, growing more forgotten with each passing year. The years between Fleming's discovery and the next pivotal moment in penicillin's history are years that haunt the medical world even today. From 1931 to 1939, penicillin existed as a scientific fact that nobody important was willing to acknowledge. It was not that Fleming's work was unknown. It was that Fleming's work was known and dismissed. His paper was published and ignored. The antibiotic properties of the mold were documented and forgotten. Fleming himself became something of a tragic figure in scientific circles, a man who had made a remarkable discovery only to watch the world pass by and forget about it. Meanwhile, during this same decade, what was happening in hospitals and on battlefields and in the ordinary lives of ordinary people? Millions were dying. Tuberculosis was claiming lives. Pneumonia was claiming lives. Syphilis was claiming lives. Gonorrhea was claiming lives. Infections from simple wounds were becoming fatal with terrifying regularity. In 1939, as the world teetered on the edge of another massive global conflict, as Nazi Germany was preparing for invasion and conquest, as millions of people faced the possibility of violence and destruction, something else was happening that would prove equally transformative. In Oxford, England, at the Sir William Dunn School of Pathology at Oxford University, a young biochemist named Ernst Boris Chain was reading through scientific literature, preparing research that might lead to grant funding and collaborative work. Ernst Chain had fled Nazi Germany in 1933, escaping the rising tide of fascism and anti-Semitism that would eventually consume 6 million Jewish lives. Chain was not a native Englishman. He was a German-born Jewish scientist who had been stripped of his position and his dignity in his homeland. He had arrived in England with little more than his scientific knowledge and his absolute determination to prove himself, to demonstrate that his brain and his talents were worth far more than any government's racial prejudice could diminish. Chain was a chemist of extraordinary skill, but he was also something else. He was a man with something to prove. He was a refugee with no safety net and no fallback option. He had to succeed. He had to make discoveries. He had to demonstrate his value. And so it was Chain who, in 1939, while reading through the scientific literature on antibacterial substances, came across Fleming's paper. The paper had been published 10 years earlier. It had been forgotten by almost everyone. It had been cited almost nowhere. But Chain recognized something in Fleming's work that the scientific establishment had somehow missed. Chain recognized potential. Chain recognized that Fleming's penicillin was not just an interesting observation about bacterial growth. It was possibly a doorway into an entirely new category of medical treatment. Chain brought Fleming's paper to the attention of his supervisor, the Australian-born pharmacologist and pathologist Howard Florey. Florey was no newcomer to scientific research. He had an established reputation as a brilliant researcher and a skilled administrator. But Florey was also something else. Florey was ambitious. Florey was driven. Florey was an Australian working in England, a man who understood that excellence was the only currency that mattered in the world of international science. When Florey examined Fleming's paper and looked at the research data Chain presented, something clicked. Florey could see what Fleming had seen nearly a decade earlier. More importantly, Florey could see what the rest of the scientific world had been too blind or too entrenched in conventional thinking to see. Florey and Chain, with the support of Florey's research team at Oxford, decided in 1939 to conduct their own investigation of penicillin. But they approached it with a rigor and a systematic methodology that Fleming had been unable to apply with the resources available to him at St. Mary's Hospital. At Oxford, Florey assembled a team of researchers. There was Edward Abraham, a brilliant biochemist. There was Norman Heatley, a young and unconventionally brilliant chemist who possessed an almost supernatural ability to solve practical problems through ingenuity and improvisation. There was Mary Ethel Florey, Howard's wife, who contributed significantly to the research. There were others, including Margaret Jennings, Jean Orr Ewing, and Arthur Gardner, each bringing specialized expertise to the collaboration. For the first time, penicillin research was being conducted not by a single scientist or a small group of researchers in a laboratory overwhelmed by other duties, but by an entire team dedicated specifically to understanding penicillin's properties, developing methods to produce it, and determining its potential as a medical treatment. The team at Oxford began where Fleming had left off. They confirmed his findings. They verified that the penicillin produced by the Penicillium mold possessed genuine antibacterial properties. But they did something else. They began to systematically address the question that had defeated Fleming for years. How could penicillin be extracted, purified, and produced in sufficient quantities to be medically useful? The problem was monumentally difficult. The penicillin in the mold broth existed in extraordinarily small concentrations. When Florey's team began their extraction attempts, they found that producing even tiny amounts of penicillin required processing dozens of liters of mold culture. The chemical instability of penicillin meant that every purification step resulted in losses. Penicillin that survived one extraction process would degrade during the next. It was like trying to capture smoke. But Florey and his team, particularly Ernst Chain and Norman Heatley, began to develop new techniques. Chain worked out methods for altering the pH of the solution and using solvents to extract the penicillin from the mold broth. Heatley, with his gift for practical ingenuity, developed the technique of reverse extraction, a method by which the penicillin could be transferred back from the solvent into water by changing the acidity of the solution. Chain suggested using freeze drying, a relatively new technique developed in Sweden, to remove water from the penicillin solution without destroying the antibiotic properties. These were not simple achievements. These were breakthroughs that required chemical knowledge, creative problem-solving, and an almost obsessive commitment to finding solutions to problems that most chemists would have considered insurmountable. By May of 1940, as Nazi Germany was launching its invasion of France, and the British people were beginning to understand that they stood alone against the Nazi war machine, as London was beginning to prepare for the bombing campaigns that would devastate the city, Florey and his team at Oxford reached a critical juncture in their penicillin research. They had managed to produce enough penicillin, through their new extraction and concentration methods, to conduct what would be the most important experiment of their lives. It would be an animal trial. 8 mice would be obtained. All 8 mice would be infected with a lethal dose of Streptococcus bacteria, one of the most deadly bacterial pathogens that plague humanity. 4 of the mice would then be treated with penicillin. The other 4 mice would be left untreated, serving as control subjects. The team worked through May 25, 1940, preparing the experiment. They injected all 8 mice with the lethal streptococcal bacteria. Then they administered penicillin to 4 of the mice. As darkness fell and the laboratory closed for the night, they had no idea what would happen. No one had ever attempted this before. No one knew if penicillin would have any effect whatsoever when administered to a living organism. Would the penicillin kill the bacteria before it killed the mouse? Would the penicillin itself prove toxic to the animals? Would the whole experiment simply prove that Fleming's laboratory observation, while scientifically interesting, had no practical medical application? They returned the next morning. Norman Heatley recorded in his diary what he saw. He wrote that when he arrived at the laboratory, he found the control mice, the ones that had not been treated with penicillin, dead. All of them. The lethal dose of streptococcal bacteria had overwhelmed their immune systems and killed them in a matter of hours. But the treated mice, the 4 mice that had received injections of penicillin, were alive. The mice appeared healthy. They showed no signs of distress. They were moving around their cages. They were eating. They showed every indication that they had recovered from the bacterial infection that would normally have been fatal. Ernst Chain, upon hearing the results, said something that perfectly captured the historical significance of that moment. He said, "It's a miracle." And it was. For the first time in human history, it had been demonstrated that a substance could kill bacteria and keep the organism alive. For the first time, a treatment existed that could prevent death from bacterial infection. Fleming's discovery had been verified. It had been validated. It was not a scientific curiosity or an interesting but useless laboratory observation. It was a potential salvation for millions of human beings. Florey and his team immediately began preparing to move forward with human trials. But here they encountered an obstacle that would test their resolve and their ingenuity in ways that no amount of scientific training could have prepared them for. To conduct meaningful clinical trials on human patients, they needed penicillin. They needed large quantities of penicillin. They needed supplies that would allow them to treat multiple patients over an extended period, to determine what dosages were effective, to understand what side effects might occur, to establish whether this laboratory phenomenon could actually translate into practical medicine. But the production capacity that had been barely adequate for mouse experiments was completely inadequate for human trials. The entire Oxford laboratory, working at maximum capacity, could produce only minuscule amounts of penicillin. The team had essentially converted their research facility into a makeshift pharmaceutical factory. But even with Norman Heatley's brilliant innovations in extraction and purification, even with the entire team working around the clock, they could not produce enough penicillin to treat more than a handful of human patients. And they were now in December of 1940. Britain was at war. The laboratory was under threat from Luftwaffe bombing campaigns. Resources were scarce. The pharmaceutical industry was completely committed to wartime production of other medications. No company could spare the resources to attempt large-scale production of an experimental compound that was chemically unstable and difficult to extract. Florey made a decision that would require courage and an understanding of political and scientific realities. He could not solve the penicillin production problem in Britain. Britain was committed entirely to the war effort. The industrial capacity was not available. The resources were not available. The expertise in large-scale fermentation and chemical engineering was not readily available. But America was not yet in the war. America had industrial capacity. America had pharmaceutical companies with resources. America had expertise in fermentation that had been developed in agricultural industries. Florey and Norman Heatley would travel to America in the summer of 1941. They would carry with them samples of penicillin mold. They would carry with them documentation of their research. They would carry with them the results of their experiments. And they would do something extraordinary. They would attempt to convince American pharmaceutical companies to invest in producing a substance that had never been made at scale, that was chemically unstable, and that would require significant investment and technical innovation to produce in any quantity whatsoever. In June of 1941, Howard Florey and Norman Heatley departed England for the United States. They brought with them something extraordinarily precious. They had brought samples of the Penicillium mold in sealed vials. But they were terrified that something might happen to these vials during the journey. They were terrified that the mold might die. They were terrified that the most important samples in the world might be lost to accident or enemy action. And so, In an act that seems almost impossibly casual in its pragmatism, they did something that would have horrified any modern biosafety officer. They smeared their coat pockets with the Penicillium mold. If the vials were lost, they would still carry the mold on their clothing across the Atlantic. The journey itself was dangerous. They were traveling during wartime. German submarines were actively hunting transatlantic vessels. But Florey and Heatley made it to America, where they were connected with the American scientific establishment and the American pharmaceutical industry. Florey had made contacts at Yale University while he was a Rhodes Scholar years earlier. Those contacts proved invaluable. They introduced him to individuals who understood the American scientific landscape. They introduced him to people who had influence with pharmaceutical companies. Eventually, Florey and Heatley were directed to Robert Thom, the chief mycologist of the United States Department of Agriculture, and Andrew Jackson Moyer, the director of the Northern Regional Research Laboratory in Peoria, Illinois. This laboratory was not primarily focused on pharmaceutical development. It was a facility dedicated to agricultural research, particularly research on fermentation processes related to corn and other agricultural products. But this proved to be exactly what was needed. The experts at the Northern Regional Research Laboratory in Peoria understood fermentation. They understood how to grow organisms at scale. They understood how to extract useful compounds from biological material. They understood the engineering and chemistry involved in taking something from laboratory scale and moving it to industrial scale. When Florey and Heatley arrived at the Peoria laboratory with their precious samples and their research data, something remarkable happened. The American scientists did not dismiss them. They did not look at the data and decide it was too risky or too difficult. They looked at the opportunity. They understood the military significance. They understood that if penicillin could be produced at scale, it could save the lives of American soldiers who would inevitably be wounded in combat when America entered the war. Moyer and his team at the Northern Regional Research Laboratory began work on penicillin production immediately. But they encountered the same chemical problems that Florey's team had encountered in Oxford. The penicillin was difficult to produce in concentrated form. The yield was extraordinarily low. The instability of the compound meant that significant losses occurred during processing. But Andrew Moyer made a discovery that changed everything. Moyer was familiar with corn processing. He knew that corn starch manufacturing produced a byproduct called corn steep liquor, a substance rich in sugars and amino acids and nitrogen. What if they used corn steep liquor as the growth medium for the Penicillium mold? What would happen to the yield of penicillin? Moyer ran the experiment. The results were staggering. When the Penicillium mold was grown in corn steep liquor instead of the traditional media that had been used in England, the yield of penicillin increased exponentially. Suddenly, producing meaningful quantities of penicillin seemed possible. The American pharmaceutical industry became interested. Companies including Merck, Squibb, Abbott, Lederle, and Pfizer began investigating penicillin production. The War Production Board prioritized penicillin research. Funding flowed from government sources. Research teams were assembled. Engineers who specialized in fermentation began designing deep fermentation tanks, massive containers in which the Penicillium mold could be grown on an industrial scale. By 1942, the first significant quantities of penicillin produced at industrial scale began to reach clinical trials. Soldiers with infected wounds were being treated with penicillin. The results were extraordinary. Infections that would have resulted in amputation or death were being stopped. Wounded soldiers were recovering. They were returning to duty. They were being saved by the miraculous substance that Fleming had observed in a contaminated petri dish 14 years earlier. But before the large-scale production was successful, before penicillin became widely available, there was a human trial that haunts the history of this discovery. In February of 1941, back in Oxford, before the American effort had produced significant quantities of penicillin, Florey and Heatley conducted the first trial of penicillin on a human patient. The patient was a 43-year-old police constable named Albert Alexander. Alexander had been pruning roses in his garden when a thorn had scratched the side of his mouth. It was a minor injury, the kind of injury that should have been trivial. But the scratch became infected. Staphylococcal bacteria entered the wound. The infection spread. It invaded his sinuses. It spread to his eyes. It spread to his face. It spread to his lungs. Within weeks, Albert Alexander was dying of septicemia, a systemic infection that had overwhelmed his immune system. His situation was hopeless by any conventional standard. He was going to die. There was no treatment available that could save him. Florey and Heatley made the decision to treat him with penicillin, with the precious, inadequate supply of penicillin that they had managed to produce through their extraction methods. On February 12, 1941, Albert Alexander received his first injection of penicillin. Within hours, something extraordinary happened. His temperature dropped. His appetite returned. The abscesses covering his body began to shrink. The infection appeared to be responding to the penicillin. After 5 days of treatment, Albert Alexander showed every sign of recovery. The infection was being defeated. The penicillin was working. But then the impossible happened. The supply of penicillin ran out. Florey and his team had produced only enough for 5 days of treatment. They attempted to extract additional penicillin from what supplies they had, including attempting to recover penicillin from Alexander's own urine, but they could not produce additional quantities fast enough. The infection, which had initially been halted by the penicillin, resumed its assault on Alexander's body. The improvements began to reverse. The fever returned. The abscesses began to recur. On March 15, 1941, Albert Alexander died. He died not because penicillin did not work. He died because there was not enough penicillin. He died because the entire world supply of penicillin was measured in quantities that could be injected into a single patient for only a few days. He died as a symbol of what could be achieved and what could be lost if penicillin production could not be scaled up. But his death was not entirely in vain. His case became famous. The newspapers in England reported on the police constable who had been treated with a remarkable new substance called penicillin. The case generated interest and attention. It demonstrated the real medical potential of Fleming's discovery. It showed scientists and physicians and pharmaceutical executives that penicillin was not a theoretical substance. It was not merely an interesting laboratory observation. It was a treatment that could actually affect the course of disease in human beings. Florey, recognizing the tragic limitations of his supply, made a strategic decision. He and his team would focus their limited penicillin supplies on treating children. Children, with their smaller body sizes, would require smaller doses. Smaller doses meant that the limited supplies could treat more patients. Children represented the greatest hope for demonstrating the medical efficacy of penicillin while being realistic about the extremely limited quantities available. And so the Oxford laboratory continued. Florey and Heatley returned periodically to report on progress in America. The Northern Regional Research Laboratory in Peoria continued its fermentation research. The American pharmaceutical companies began constructing the fermentation tanks and engineering systems that would make mass production possible. And by 1942, the first significant quantities of penicillin produced at industrial scale were beginning to reach the military. During the D-Day invasion in June of 1944, American soldiers landing on the beaches of Normandy had access to penicillin. They had access to a treatment for infections that no soldier in any previous war had ever possessed. Wounds that would have meant amputation in World War I could now be treated. Soldiers could recover. They could return to combat or return home alive. But the story of penicillin production was not limited to what was happening in Peoria, Illinois. In 1942, as the American war effort was shifting into higher gear and pharmaceutical companies were racing to produce penicillin at scale, the Northern Regional Research Laboratory in Peoria began a global search for Penicillium strains that might produce higher yields of penicillin. They understood that Fleming's original strain, while productive, was not necessarily optimal. They sent soil samples requests around the world. They received samples from every continent. Scientists hunted for fungi in diverse environments, searching for the rarest and most productive strains of Penicillium. But one of the most productive strains would be discovered in the most mundane and ordinary place imaginable. Mary Hunt was an assistant at the Northern Regional Research Laboratory in Peoria. She was a woman working in a facility that was almost entirely male-dominated, a scientist contributing to research that would shape the course of medical history. One day in 1942, Mary Hunt visited a local market in Peoria, Illinois. She was shopping for groceries. In the produce section, she noticed a rotting cantaloupe melon. The melon was spoiled. It was moldy. Most people would have walked past without a second thought. But Mary Hunt recognized the significance of what she was seeing. She examined the mold. She collected a sample. She brought it back to the laboratory. The mold on that spoiled cantaloupe melon, the mold that Mary Hunt had found while shopping at a market, proved to be a strain of Penicillium chrysogenum that produced 6 times more penicillin than Fleming's original strain. This discovery accelerated penicillin production exponentially. By 1943, American pharmaceutical companies were producing penicillin in quantities that would have seemed like fantasy just 2 years earlier. By 1944, production had reached levels that could supply the needs of Allied military forces. By the end of World War II, American pharmaceutical companies were manufacturing 650 billion units of penicillin every single month. 650 billion units. The substance that Fleming could barely produce in microgram quantities just a decade earlier was now being produced by the ton. Fleming himself shared the 1945 Nobel Prize in Physiology or Medicine with Howard Florey and Ernst Chain. The 3 men recognized for revolutionizing medicine by bringing penicillin into the world. By the 1950s, penicillin had saved an estimated 200 million lives. The simple contaminated petri dish that Fleming had almost discarded had led to a cascade of discoveries, innovations, and developments that fundamentally transformed human civilization. Infections that were previously fatal could now be treated. Soldiers could survive their wounds. Mothers could survive childbirth. Children could survive bacterial infections that would have been universally fatal in previous generations. The world had been changed, and it had come so close to being lost forever. All because one scientist, exhausted and frustrated, took a moment to look more closely at something that should have been discarded as contamination. Before we finish this incredible journey through history, I want to ask you a question that might be haunting you right now. How many other discoveries like penicillin have we missed? How many other contaminated petri dishes have been thrown away? How many other researchers gave up too quickly? How many other opportunities for breakthroughs have been lost because people were not paying close enough attention? The story of penicillin is not just the story of Fleming or Florey or Chain or Heatley or all the brilliant scientists at the Northern Regional Research Laboratory who worked tirelessly to solve an impossible problem. It is a story about human persistence. It is a story about collaboration. It is a story about the importance of paying attention to things that do not fit your expectations. It is a story about the possibility that something mundane, something that looks like failure or contamination or accident, might actually be the key to saving millions of lives. Thank you for watching this journey through one of the most important discoveries in human history. If you found this story compelling, if you felt the weight of how close we came to losing this miraculous substance, Please share this video with someone you care about. Leave a comment telling us what you think would have happened if Fleming had simply cleaned up his laboratory that day. Subscribe to our channel so you never miss our exploration of the hidden stories behind humanity's greatest breakthroughs. The story of penicillin is finished, but the story of medical discovery continues. And we want you to be part of that journey with us.